Welcome to our detailed guide on gram staining, a fundamental technique in microbiology used to differentiate bacterial species into two groups, gram-positive and gram-negative, based on the characteristics of their cell walls. Let's start with a step-by-step demonstration of the gram staining procedure. First, prepare a bacterial smear on a clean glass slide. Allow it to air dry and then heat fix by passing it through a flame. This step ensures the bacteria adhere to the slide and are killed. Apply crystal violet stain to the smear and let it sit for 1 minute. This primary stain will initially color all the cells. Rinse the slide gently with water to remove excess stain. Next, apply iodine solution and let it sit for 1 minute. Iodine acts as a mordant, forming a complex with the crystal violet, making it more difficult to remove from gram-positive cells. Rinse the slide again with water, then apply a decolorizer, such as alcohol, for about 10 to 20 seconds. This step is critical. It will remove the crystal violet iodine complex from gram-negative cells, but not from gram-positive cells. Rinse the slide with water once more and apply safranin for 1 minute. This counterstain will color the gram-negative cells pink, while gram-positive cells remain purple. Rinse off the safranin, blot the slide dry with bibulous paper, and examine it under a microscope using oil immersion. You should now be able to distinguish between gram-positive and gram-negative bacteria. Gram staining differentiates bacterial species into two groups, gram-positive and gram-negative, based on the characteristics of their cell walls. Gram-positive bacteria have a thick peptidoglycan layer in their cell walls, which retains the crystal violet stain, appearing purple under a microscope. In contrast, gram-negative bacteria have a thinner peptidoglycan layer and an outer membrane, which does not retain the crystal violet stain, but takes up the counterstain, safranin, appearing pink. This differentiation is crucial for the identification, classification, and determination of appropriate antibiotic treatments for bacterial infections. Let's discuss some common mistakes that might alter your results and how to avoid them. Over-decolorization can result in gram-positive bacteria appearing gram-negative, while under-decolorization can cause gram-negative bacteria to appear gram-positive. Ensure proper timing and technique during decolorization. Improper heat fixing can distort bacterial morphology. Pass the slide through the flame briefly and evenly to avoid overheating. Consistent timing for each staining step is essential for accurate results. Set a timer to ensure each stain is applied for the correct duration.